Hey everybody, welcome back to Microbiology Bio229 at HCC. I'm your host, Mary Annarella, and we are finishing up chapter 13 today, which is all about viruses. I left the little U out of viruses. How about that? Viruses and viral infections. All right, we have already talked about the lytic cycle. We have talked about latent viruses that become a provirus. If you don't know what that sentence means, you need to go back and look in your notes and figure out what a provirus is. But a lot of viral infections are not lytic. Some of them are temperate. So we're going to be talking a little bit about different types of viral infections in humans. So to do that, let's just take a look at, oops, wrong, my bad, how about that? All right, this is what we're talking about. Types of viral infections. Viral infections can be acute, meaning short-lived. With an acute infection, a person will get infected with a virus. Their immune system will clear it, and it doesn't come back. So, for example, the flu is like this. The flu is an acute infection, but there are viral infections that persist, meaning that there's always virus present somewhere in the host cell, and depending on the type of persistent infection, a person might be contagious. Or they might have periods where they are not contagious at all. I will say for acute infections, which may or may not develop lasting long immunity, okay, the flu is an example of that. Also COVID. COVID is an example of an acute viral infection. When a person is first tested for COVID, and let's say that they test positive for it with a PCR test, with the antibody test. They get sick for a while. At some point, even if there are lasting health effects, their body is cleared of the virus. They will eventually have a negative COVID test where we won't be able to detect any of the virus, any viral gene sequences, with PCR. So they clear it. They don't hold the virus with them. They're only contagious for a certain period of time after they have had that infection. That is not the case with a lot of infections. So let's delete that screenshot there. Okay, but some are persistent infections. So let's talk about this. I'm going back here. And you know what? Let's do a little graph here. It is so weird to be using black ink. I hardly ever go for that. But let's time going this way. I don't know, in days, whatever. And let's put symptoms. Symptoms and viral numbers. All right, so let's say, actually no, symptoms is not going to be on there. This is going to be the number of viral particles here. I'm not liking this graph like this. At some point, a person is exposed. The virus gets into their cells and viral numbers come up, right? And at some point, the viral numbers will taper off and you will have symptoms. This will be the symptomatic period here. Symptomatic period of time in which you have symptoms. All right, but you still have high viral numbers here and you can be contagious, right? So this whole entire area up here, this time, you're contagious here. You're contagious during this time period too. Come back. There we go. That's what I wanted. So you're contagious. Even after symptoms subside and viral particle numbers come down, you're still contagious, right? And it's usually for like 14 days after you've had a negative test is when, we let people come back and be around people. They don't have to quarantine or isolate anymore. Okay, but that's it. Like, numbers have dropped up. There's no more virus. No more virus present in this person's cells, and they are not contagious at all. So that's an acute infection. Acute. It's a one-shot deal. Persistent. Is that an A? Is that an A? I think that's an E. Persistent. No, I think it's an A. Great. I don't even know how to. No, it's an E. Dang. It is an E. Man, that just does not look right to me. Persistent infections. Even when viral numbers come down, you will still harbor the virus within you, but there are different types. There are chronic persistent infections and latent persistent infections. In the PowerPoint, I call one of them slow. We don't use that terminology anymore. I did not update my PowerPoint, and I'll go back to the PowerPoint and have you X things out. But a slow infection is the same as latent. So we'll talk about that in a minute. But let's look at latent. Latent infections. With latent infections, you get, let's draw the same graph, time over here, viral numbers over here. You get an initial infection, viral numbers come up, and you'll have a symptomatic period of time, right? And the viral numbers will come back down. But unlike an acute infection where there's no more virus, a virus that displays a latent infection hangs out in your cells. So even though viral numbers are down, here, virus is still present in some cells. And usually they hang out in your cell as a provirus. Usually as a provirus. That's when viral DNA gets integrated into the host cell. So that later on there is some sort of reactivation of the virus that's already present in your cells, and viral numbers will come up again. You'll have a periodic symptom, You'll have a period of symptoms, and then viral numbers will come back down again. An example of this, and I bet everybody here is familiar with the chicken pox, which is a varicella virus. The chickenpox virus, varicella, hangs out. You get an initial infection and you get the chickenpox. And after the blisters heal, you're not contagious anymore. But the chickenpox virus, varicella, hangs out in some of your nerve cells as a provirus. And later on in life, there is often some sort of reactivation of the virus. Those numbers come back up, and only with the chickenpox virus, with varicella, you don't get the chickenpox again. Do you know what it's called when the chickenpox virus is reactivated later? It's called the shingles. We have a shingles vaccine, and it's somewhat effective for preventing reactivation, but it doesn't prevent you from getting the virus again because you already have it in your cells. Chickenpox is an example of a latent infection. It persists. The virus is still present in some cells, period. Chickenpox, herpes is like this too. Herpes simplex. 1 and 2. You know, there are two different types of herpes. One is, let's see, herpes simplex 1, HSV. HSV1 is oral herpes. Cold sores. Cold sores. Whereas I think 2 is genital herpes. But people will have flare-ups. So for example, you know, if you get an initial infection of herpes simplex 1 and you get cold sores, you get a cold sore here, you're contagious a little bit before you get that cold sore, right? And you never know when that's going to be. And you're going to be a little contagious afterwards as the blister is healing. Later on, you get some sort of reactivation of the virus, and with cold sores, herpes simplex 1, for example, it's usually, you know, a cold or a fever. That's why we call them cold sores, or sometimes they're called fever blisters. But you get some sort of illness, your immune system is down, that reactivates the virus, viral numbers come back up, and you get another cold sore again. And that's also how it works with genital herpes, only except of having a sore on your mouth, you have it on your genitals, and that's why it is a big deal to protect yourself and have protected sex if you're going to have intercourse with someone who is infected with genital herpes. People also have been known to get genital herpes of the mouth, and I'll let you all just think about how that would happen, but protection would be required for that too. And again, I'll let you think about how you'd have to protect yourself from that. Okay, so those are latent infections. There is some non-infectious period here. But someone is going to be infectious, like right before reactivation. So we don't really know. You can't just say, oh, well, if this person has symptoms, then if this person doesn't have active symptoms, if they don't have active herpes, then they're not infectious. Not necessarily the case, because they could be somewhere on the slope of this line here before symptoms occur. So practicing safe sex in that regard would be very important to prevent transmission of herpes. All right, there's a different kind of persistent infection that is called chronic. And this would also include slow infections. In chronic persistent infections, infectious virus is always present. It's always present, whether a person has symptoms or not. So not the case up here. There is some period of non-infectiousness in latent infections, but you are never not contagious when we're talking a chronic viral infection. So chronic viral infection, I'm getting tired of this black. Let's do with something a little easier on the eyes. Let's go with brown. Oh yeah, that's so much easier on the eyes. So viral numbers over here and time over here. With a chronic infection, a person will get an initial infection. Viral numbers will come up. There will be a period of symptoms. A person has symptoms. But viral numbers are always present. Infectious virus is always present. So for example, hepatitis is an example of a chronic infection. A person who has been infected with one of the hepatitis viruses. Usually hepatitis B is the big one. There is hepatitis C as well. There also is hepatitis A, but that one's a slightly different. People with hepatitis are always infectious. And so if someone has hepatitis, and even if they don't have symptoms, if their blood is being drawn, they have to be extra precautions have to be taken, right? Because it's exposure to blood that can cause transmission of hepatitis, for example. So if you're a phlebotomist and you're taking blood from a person who has hepatitis, some extra caution has to be taken. And it's also the case with HIV. HIV we used to consider a slow infection, but it's basically the same concept as chronic. It just took longer for viral numbers to reach a critical mass to cause symptoms with an HIV infection, but really it's a chronic infection. And people with HIV, we consider them never to be rid of the virus. Even with those protease inhibitors that I talked about, extreme caution is still required. So even if they can't detect the virus, we still treat patients with HIV as though they are always infectious. So they always have to be careful. So let's see. Let's go to our PowerPoint. All right. So we talked about latent. There is an asymptomatic period of time. I think I misspelled asymptomatic. There's only one S there, pretty sure. Reactivation of the virus results in symptoms. These can be, DNA or RNA virus. Herpes simplex is an example of this, and so is chickenpox. And we had chronic. Hepatitis B is an example of that as well, but infectious virus is always present in the host, always. Not the case with latent. And then again, slow. Let's just take a picture of this. Persistent infections, slow. There, it's same as chronic. Just HIV is a slightly different caveat because you need to reach a critical mass of viral particles before developing acquired immunodeficiency syndrome. So we used to consider it a different type of infection, and we no longer do. So I need to update my PowerPoint now, don't I? Alrighty. Okay, we already talked about classifications of viruses last time, and this slide here already talked about that. We talked about retroviruses as well, of which HIV is one of them. No. Why is it doing that? Okay, doesn't want to play nice. Going on to the next one, I wanted to say a few words about viruses and cancer. There are very few viruses that attack humans that cause cancer. Very, 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 very few. HIV even inadvertently causes one. So let me take a picture here. HIV causes a type of cancer called Kaposi's sarcoma. And this, if you remember the movie with Tom Hanks called Philadelphia, it's about a lawyer who was fired from his job for having AIDS, and this was at a time when AIDS was branded as a gay men's disease. We know that the virus does not care what your sexual orientation or gender happens to be, gender or lack thereof. And so he hires Denzel Washington to be his lawyer and sues his former employer. And part of what caused the jury to agree with Tom Hanks's character and Denzel Washington's character is that he had these Kaposi's sarcomas all over his chest. And there is this scene in the courtroom where they were saying, his character was saying that they had noticed some of the Kaposi's sarcomas on his face, and that's how they figured out that he had AIDS and fired him because of that. But at the time of the trial, he did not have any on his face at all. And these are large lesions. I'll let you Google Kaposi's sarcoma, KS, if you want. But they weren't on his face, and Denzel Washington's character asks him, do you have any of them on your body now? And he said, well, I do. I have them on my chest. And and Denzel Washington is like, request permission to ask the witness to, remove his shirt. So he lifts his shirt up and everyone in the courtroom just winces. And that is kind of a deciding factor in everyone realizing that, yep, that this company had discriminated against Tom Hanks's character. Anywho, lovely little aside there. But HIV did not directly cause those Kaposi sarcoma. What it does is it represses the immune system. And it's the repression of the immune system that allowed Kaposi sarcoma to even happen, to allow those cancer cells to proliferate, because normally those cells would have had their DNA fixed by mismatch repair, for example, or better proofreading by DNA polymerase. But because the immune system didn't remove those cells, they were allowed to grow unbridled. So it's the repression of the immune system that allows HIV to indirectly cause Kaposi sarcoma. But the one big one, and you can see it sitting right here, is human papillomavirus. Human papillomavirus directly causes cervical cancer. It is directly linked, and it does not do that by suppressing the immune system at all. It does so because human papillomavirus has what we call oncogenes. They are genes that cause cancer, and they get inserted into the genome by viruses. So an oncogene, think of it as a cancer-causing gene. And human papillomavirus here, HPV, that is now covered by the Gardasil vaccine. And we talked briefly about the Gardasil vaccine, oh gosh, a month or so ago. But that is one of the few viruses that actually causes cancer. Really, really interesting. So let's see, what do I have up here? Fewer than 10 human cancers. This is the big one right there, HPV. But I'm putting that out there because a lot of people are like, oh my God, viruses cause cancer. Like, no, actually, they really kind of don't. In mice, there is something called the mouse mammary tumor virus, but we have not found any virus that even remotely is linked to breast cancer in humans, for example. So there is no human mammary tumor virus that we have found, and we've been looking for evidence for it for 50 to 60 years at least. So that is, okay, up. So that's the deal on viruses and cancer. However, viruses are not all horrible things. We can actually use viruses to our advantage to improve human health, and there are some ideas that have been bounced around over the years, so I thought I would give you an idea of how these work. Gene therapy. Gene therapy is replacing a defective gene with a healthy gene. We would use CRISPR now. We're hoping to use CRISPR now to do that. But people are like, we need a way to get a healthy gene into human cells. Like, let's say, here's your cells right here. And here are all your little chromosomes here. I'll do them like this. Look, there's the centromere. And let's say this cell is lacking a healthy copy of some gene. What other color could I use? And here's the healthy copy of that gene, and we want to somehow get it in there and hope that it recombines by homologous recombination. Again, we would use CRISPR for this now, so this is just an older idea. It's like, well, how are we going to get it in there? Because most pieces of DNA are just going to get chopped up before they get in there. I know. We'll package this good copy of gene Z, whatever, inside a virus. Or rather, just inside the shell of a virus. Just the envelope. Like, let's say this is just the envelope of a virus we know that attacks this particular type of human cell. And then the whole thing will get in there. And if we have sequences that allow this thing to become a provirus, we might possibly be able to put in a good copy of a gene to replace a bad copy of a gene and say, oh, look, there, it goes in there. Well, again, CRISPR is going to be so much more efficient at doing that, and they actually have with sickle cell anemia. But, oh gosh, it was like the first time they tried this, it was to try and cure SCID. SCID stands for Severe Combined Immunodeficiency Syndrome. People with no immune system, like people who end up having to live in a bubble, basically. And I'm not talking about a social bubble. I'm talking about a literal bubble. So if you've ever seen Seinfeld, there's an episode about that. But anyhow, so they tried to use just the envelope of a virus to get that in there, but it ended up inserting in the wrong place, and it ended up causing all kinds of problems. So this didn't work out. But I want to point out that when you use like the shell or just the envelope of a virus to get something into a cell, we would call this a viral vector. And that's going to be important when we're talking about vaccines very soon. So a viral vector. A vector is something that carries something from one place to another. So, think about raccoons. Or no, better. Think about Lyme disease. Y'all know that Lyme disease is caused by a microbe, caused by a bacterium that is carried by deer ticks, right? Deer ticks carry the infection basically from the deer there's the deer, and the deer tick bites the deer, picks up the infection, and then the deer tick bites you. Gosh, it sounds like one of those jokes. In Russia, the deer tick bites you, or whatever. You bite deer tick. I don't know. That's a really bad, stupid joke. Anyhow, the deer tick carrying the microbe. Oh, we need to put the little green microbe on there. The deer tick is called a vector. Vector. And the virus can also be a vector because it's carrying that gene into your cells. So it's called a viral vector. This is also kind of sort of how a lot of vaccines, including two of the promising vaccines from Moderna and Pfizer, they're using a viral shell. They're using the envelope. So really, they're using a viral vector to get that messenger RNA into your cells. So that's an interesting thing, and I just want to talk briefly about gene therapy. So far, it hasn't worked. But CRISPR looks very, very promising, and they've actually used it to cure someone, to give them a healthy copy of a hemoglobin gene into someone who had sickle cell anemia. So it's not going to work except through CRISPR now. Prions are another interesting thing. Prions are proteinaceous infectious agents. You've probably heard of spongiform encephalopathy by another name, mad cow disease. It's a degenerative disease of the gray matter in your brain. It's considered to be a slow infection because it takes a very long time for these prion particles to build up into sufficient numbers so that they cause disease. But oh, that's right, I have to get out of there. So let's talk prions. They're proteins. It's a protein, rather. It's a protein that normally exists in humans and also other animals. Let's see. It's a it's a protein in mammals. And I'm saying mammals because it's common in bovine, it's common in sheep. What is sheep? Is that, no, that's not ursine. Sheep is not porcine. I forget what ein that sheep are. Anyhow, it's in mammals, and it's a normal protein. The normal version, the normal, let's say healthy version, let's call it prion protein cellular. That C is for cellular. And let's say, here it is. Here's our little globular protein. There exists a version of it in what's called scabies. There's a disease called scabies in cattle and sheep that looks exactly like the normal prion protein, but with a couple of differences. So the prion protein, we're going to put SC, and this stands for scabies. So that's no, I don't want to use black, purple. Here we go. PRPSC. What is so weird is that the messed up version, the prion protein, the scabies version, and the normal version have the same amino acid sequence. Weird. Really weird. But they have a different shape. Totally. Different shapes. And the way it works is, let's go back to, okay, brown is the normal healthy version, purple is the one that causes disease. Let's write that down. Scabies. This one causes disease. The way people can get mad cow, or that all sorts of these spongiform encephalopathies can occur, is that, okay, let's see, here's a cell, and here's your normal prion protein right here, hanging out, minding its own business. Let's say the owner of this cell, this organism, eats tainted meat that has some of the scabies prion protein in there. This gets into the cell, and what is so weird, and we don't know how this happens. Now remember, this has a different shape from this one, even though they have the same order and number of amino acids, right? There is something about the interaction of this purple one, the abnormally shaped one, to this one. They will have some sort of contact with each other, and that contact converts the healthy brown one into a disease-causing purple one. And now this one's going to go on, and it'll have contact with this one and turn this one into a disease-causing version. It's a slow infection because as they continue to have contact with each other, the mutant numbers build up. And again, we don't know how this works, but once enough of these numbers build up, then disease happens. But there is something about contact of the misfolded protein with a normally folded protein that causes the normal one to fold in the wrong way. It is really, really weird. And what it does is that it messes up this whole, you have your DNA, which copied into RNA, into protein thing, And then if you're going to get a different protein, you need to have a different gene for it back here. And this messes up the whole central dogma of biology. It's freaking weird. When people were studying this back in the 80s, people thought people studying scapies, prions, were absolutely freaking nuts. Like, how could you possibly have two proteins that are the same exact amino acid sequence and have this one fold differently from that one? It makes no freaking sense. And even less sense is the fact that the contact between the two causes the healthy one to misfold into the shape of the evil one, into the shape of the disease-causing one. It's freaking weird. People thought these researchers were nuts. Let me show you this nice little graphic here. Oh, okay, I used different colors. Let's do this right here. But here we are. I just pulled this off of Wikipedia because it's a really nice, easy graphic. Okay, so here's the healthy one up here, all these green ones. They're just minding their own business. They're not bothering anybody. But let's say this person, this organism ate tainted meat that had the mutant in there. And here we're saying, it could be spontaneous generation of them. Inoculation, that means eating contaminated meat. This bad boy gets into an organism's cells. There is something about the interaction between the two of them that causes the good one to convert to being the mutant one. It's like, what is this like? It's like these little green ones are Anakin Skywalker, and the bad one is, well, not Darth Vader, that doesn't work, not Darth Maul, the Emperor Palpatine. Emperor Palpatine's the bad guy who's on the dark side, and Anakin was so innocent and so nice for so long, but no, he had to get lured over. Just being in the presence of the evil emperor lured Anakin over to the dark side, and he is Darth Vader. I don't know how to draw the Darth helmet, whatever there. That is really bad. That looks nothing like Star Wars there. But now he's Darth Vader, right? And we'll try to and we'll actually try to go and convert Luke Skywalker. And after that, the analogy breaks down big time. So I think we'll move on here. There we go. What are we doing here? Back to prions. Okay, so they are a slow infection. One other thing I wanted to add about prions is that they are so freaking heat stable. 20 minutes in an autoclave is not going to cut it. I think it's something like it needs 2 hours at high temperature and pressure, not 20 minutes. So it's kind of crazy that it requires all of that. Alrighty. So that basically finishes up. Oh, diseases caused by prions. Yeah, mad cow. They're all forms of spongy encephalopathy. This is just to point out there are a couple of other diseases caused by the prion protein. One is an extreme familial insomnia. Around age 35, people who have this are not able to sleep and end up dying from that. Kuru is a laughing disease, and it comes from consuming contaminated meat, and it's only found in some cannibalistic societies, which I hope to God there are no more left anymore that anyone would do that. But they do exist, and actually these are due to, oops, wrong one. Actually, it's the familial insomnia that is due to a faulty gene. So there are actually a few spongiform encephalopathies, a few things caused by prions where it's not, the whole Darth Vader trying to convert Luke Skywalker thing, but rather due to a faulty gene. Delete that. Okay. Yeah. That was the only thing there. You can get a spontaneous mutation, ingestion from contaminated meat source, and this is why if they find any evidence of prion proteins in mad cow disease in a herd of cattle, the entire herd of cattle have to be destroyed. Because if that meat is contaminated, it can and will lead to spongiform encephalopathy, mad cow disease. So that finishes up this weird thing is going on here. I'm going to close that photo up from Wikipedia. Where'd we go? Notability. That looks weird there. That gets us into chapter 14. Chapter 14 and 15, I am taking these together. So this is all about innate immunity and the immune response, or rather, and the application of adaptive immunity. It's working on it. Here we go. Oh my God, we're almost done. We are in the home stretch. We do not have that many more lectures before the end of classes. So that's very exciting. And I'm especially excited that we get to be talking about immunity. You will note that, or you will notice, hopefully, and I will talk to you about this in the Zoom class, but I have an article for you to read. It's from the New York Times. It's old, but it's still very, very relevant. There is an assignment there for you. You need to read it and then type up 3 points about it. Each point should be 2 to 3 sentences or so. So that's up there for you there, and it is related to what we're going to be talking about for the rest of the semester, which is immunity. 2 different types of immunity we're going to be talking about. First up, there is innate immunity. The word innate means a part of your system already. Systems that are already in place that will allow us to recognize a pathogen when we see it. So we'll be talking about those. This is different from the adaptive immune response. This is when your body is presented with a new microbe, your immune system learns to recognize it so that if it sees it again, it will be able to fight off an attack, right? Like you're not going to get the measles twice. You're not going to get the chicken pox, you're not going to get the chicken pox twice. It's going to manifest again. It's going to be from the same infection. You can't get the same infection twice. That's if you have an adaptive immune response. So we're going to take, what is my dog doing? She's trying to hop in or out of her little doggy basket. Okay, I'm waiting to see if she needs to trot outside, but anywho, we're going to talk about innate immunity first. Here we go. These are your first lines of defense. Okay, so your skin is one of your defenses. A microbe has to get in you if it's going to infect you. So your skin and your mucosal membranes, they are not the same thing. There are also antimicrobial substances that your body already knows how to make. You have the genes to make these substances already, and you do. And then, of course, there is the normal flora, and that's what that New York Times article is about, and we will discuss that very soon. But first up, skin and mucosal membranes. So the skin keeps the outsides out, the insides in. You have several different layers to your skin. So your epidermis is what's up above, and your dermis is below that. And so let's see. You'll have, I really don't want to do it that way. Here, yellow, yellow, yellow is what we're after. All right. There we go. All right, so down here in your dermis, this is mostly made up of fibroblasts. I'm not doing a very good job of drawing fibroblasts, but they're going to be somewhat brick-like, somewhat rectangular, but they're going to be lengthy. And this is what we call stratified epithelium. And I know there are several different types of epithelium, and those of you taking A and P could probably rattle them off much more easily than I could. All right, so your dermis is actually vascularized, so it has blood vessels. And just above your dermis, separating the dermis from the epidermis, is what we call the basement membrane. And you will have all of these cells here. Basically, nutrients need to diffuse into your epidermis. And the layers get thinner and thinner, and the dead cells slough off like that. But in there you will have some basal cells. You'll have keratinocytes. They're the ones making keratin, which actually forms a keratin is a protein that's in hair and nails. It's also in your skin, that provides a really nice barrier. This is a good barrier protein. Your melanocytes produce melanin, and that's what will determine the color of your skin cells too. I have way more melanin than my mother does, for example. Not as much as my dad does, because he's just got more melanocytes than I do. Okay, but it's stratified epithelium. Think of skin as literally a wall. It's a physical barrier. And the keratin in the outer layer forms kind of a waxy barrier so that most things can't get in unless you introduce a cut. Like that. Your mucosal membranes don't have stratified epithelial. They have columnar. And yes, I know there's cuboidal and all this other stuff. We are just sticking with a couple of them. So columnar, they're going to be in columns instead of horizontal bricks. They're often ciliated, so they'll have cilia. So think of these as like upper respiratory tract. And the cilia help beat away a particle or a pathogen. Mucosal membranes also secrete mucus. Not a big surprise. You have mucus in your upper respiratory tract. You have mucus up in your nose. Alrighty, so that's columnar epithelium. So mucus can actually have some antimicrobials in it, but it also just provides more of a barrier as well. And it's movement of the cilia that can help beat away a pathogen. So that's, again, a first line of defense that a microbe has to get through in order to cause an infection. Next up, your body naturally produces antimicrobial substances. So, for example, you produce lysozyme in tears, and also in saliva. Saliva tends to have lysozyme in it. And if you remember this, I have up here not as effective against gram-negative bacteria, because who can tell me why? Can you think about that? What do gram-negative bacteria have that gram-positive bacteria don't? It's like, oh, gram-positive bacteria have this really thick layer of peptidoglycan. So lysozyme can't get through that, right? Wrong. No, it can get right through there. No problem if it's gram-positive. Gram-negative has a thin layer of peptidoglycan, but they also have this outer lipopolysaccharide membrane out here. Bam, lysozyme can't get through that. So lysozyme in your tears and in saliva will be somewhat effective in killing off some gram-positive bacteria, but not gram-negative bacteria so much. We make a lot of peroxidase enzymes that can destroy bacteria, but if bacteria are catalase positive, they'll just break that up, so that's kind of a problem. Lactoferrin binds up iron that is often required for a lot of microbes to establish an infection. Lactoferrin, what does that sound like? It's in breast milk. So there are lactoferrins in breast milk. I put stomach acid here as an antimicrobial substance because stomach acid is a wicked low pH. It's super acidic, and it will destroy a lot of bacterial cells. It won't destroy toxins that you ingest, though. So if a bacterial cell like bacillus is on your food and it has produced some spores or it has produced a toxin, your stomach acid isn't going to break up that toxin. But stomach acid can definitely kill several different types of bacteria. A lot of them. All right, and then there is the normal flora. Oh, the normal flora is so wonderful. What would we do without it? You all know that our bodies are not sterile, right? The moment we're born, we start establishing a population of bacteria all over our bodies. They live on the body's surface, and sometimes, as you're going to find out from this New York Times article, inside the body too. And I'm not just talking your lower GI tract. Okay, you know that your skin is just loaded with bacteria and some fungi as well, yeast and fungi. It's their home. They belong there. You don't want to get rid of them because they do a really good job of scaring away pathogens. And what I mean by scaring them away, they compete with pathogenic microbes. So, for example, they compete with pathogenic microbes for nutrients. They compete with pathogenic microbes for basically real estate. And by real estate, a place to set up shop. Let me get rid of this and talk about it from this point of view. Okay, well, my iPad crashed, and I don't know what the heck happened there, and this is really, really weird. But let's say here are your skin cells. Darn it, I shouldn't have gotten rid of that stratified epithelium. Okay, now I'm going to do a crappy job of drawing the dermis because I'm in a hurry here. All right, let's draw in the basement membrane. Cells are getting thinner, and they'll slough off eventually. Let's say green is the color of microbes. We have lots of microbes living all over your skin. Lots of bacteria, tons. Oh gosh, staph, strep, coliform bacteria is everywhere. And yeast would be a little bit bigger. I'll draw it here. You see the space they're taking up? Now, what if a pathogen comes along? Like, haha, I am an evil bacterium. It's like, can I move in here? And everybody here is like, nope, there's no room for you. Sorry, got to look elsewhere. And even if they do manage to say, I'm going to squeeze in here, they're probably not going to have much nutrients, right? Because all these green guys, all the flora is already munching down on all the nutrients that are there for it. So one of the ways they do that, they just crowd everybody out. Okay, did this crash too? This may have crashed. Oh, do work on it. Thank you. Welcome back. 2 minutes ago. All right, where are we? All right. They compete with pathogenic. Oh, well, the last screen recording video saved. That's good news. They compete with pathogenic microbes for space. They compete with them for nutrients. Antibiotics can screw up your flora big time. They can cause diarrhea because they kill off all of the microbes, or kill off too many of the wrong microbes in your gut. A lot of times a lot of women will go on antibiotics for, I don't know, say something like a sinus infection, although drugs have gotten a little bit better now. You end up with a vaginal yeast infection because the antibiotic has disrupted the balance of different microbes in the normal flora of the vagina. That's definitely a thing. And you end up with a yeast infection, and you have to take an antibiotic to get rid of it. There is antibiotic-associated colitis. People end up with really bad irritable bowel syndromes or unable to digest a lot of different foods because they were on antibiotics that were so strong that they killed off way too many of the microbes in their lower GI tract. And actually, people have done fecal transplants. And as Stephen Colbert once said on the Colbert Report, it's like a heart transplant, but with poop. It literally is. It's helping. Fecal transplants, they literally take microbes isolated from the poop of a healthy person, and they actually sometimes will take a scope and go through the mouth and the stomach so that they can deposit it in the small intestine to get it into your colon to hopefully repopulate the colon and lower GI of someone with this antibiotic-associated colitis. Also, C. diff infections can result in that. Clostridium difficile. If any of you work in healthcare, and I know that a lot of you do, you are probably well aware of Clostridium difficile and the havoc that it can wreak on the hospital environment and how contagious it is. But people can get severe colitis from it, and I don't know how widespread this whole fecal transplant is. It's relatively new in the past 10 years, but that is the concept. So don't mess up your flora. There's one more thing that I want to talk about what the flora do. Not only do they compete with pathogenic microbes for living space and for nutrients, there is also, I can't do that. When am I going to learn this? Here we go. There is also evidence that the normal flora, we need green now. I guess I chose green for the color of the normal flora. Normal flora also have been shown to be involved in the human immune response. Where they can work with antigen, where they basically can summon the immune system to come on over here. Not quite like an antigen presenting cell, but they will recognize a pathogen and give a heads up to the human immune cell. I mean, to the human immune system. There are way too many cells of the human immune system. But they coordinate their activities with human immune cells, with B cells, T cells. So you will read more about that in the article that I have linked for you to read sometime during the week of Thanksgiving. It will be due the Tuesday after Thanksgiving. We will talk about it in Zoom. And next time we will pick up and we will talk more about the adaptive immune response. I hope you have a great day. And I can't remember if you're going to be watching this before or after Thanksgiving. I think this is actually, you're listening to this before Thanksgiving. So I hope you all have a lovely Thanksgiving and a very safe one too.